Same agent, a single-cell request. BioMate routes to the OSCA stack, clusters and annotates in parallel, then runs condition DE the statistically correct way, pseudo-bulk. 7 phases: QC, normalize, feature selection and PCA, cluster, then markers and annotation in parallel, then pseudo-bulk DE. The agent works backward from the goal. Condition DE with DSEQ2 means a single-cell experiment plus scRAN and scatter, not Seurat, so the final step hands off cleanly. Phase A. Empty drop separates real cells from ambient barcodes, then per-cell QC flags, high mito, low-count cells. 9,814 of 12,402 kept. Each step opens with defaults filled in. Click the gear to adjust a value inline. Change only what you need. Every phase has an editable QC gate, pre-filled with its default. The auto loop switch lets a failed gate fix its params and rerun. With auto loop on, a failed gate posts a halt box. Failing metric, proposed fix. 10-second countdown. Apply, pause, or dismiss. Files drop into the panel as each step finishes. View opens them in line. Download grabs the file. Any phase can be stopped. Finished steps are kept. Then resumed where you left off. Nothing recomputed. QC and normalization fold into a single-cell experiment carrying counts, log counts, and metadata. Every step below reads from this one object. Normalization, HVG selection with PCA, and graph-based clustering are done. 11 clusters. The UMAP is for visualization only. Clusters come from the SNN graph. Phases E and F open together. Score markers and SingleR both read the clustered object, so the engine fans them out and tracks the join. The key beat, testing thousands of cells as replicates, is pseudo-replication. The fix, aggregate to pseudo-bulk, so the 6 samples are the real replicates, then DCQ2 per cell type. 7 phases done, routed to OSCA, clustered, annotated, then pseudo-bulk DE. CD8 T cells are the strongest responder, 214 DE genes on true replicates. The after-run report, 7 tabs in reading order, results first, audit last, and a one-click export of the whole document. Findings, headline plus figures from the manifest, DE genes per cell type, composition, QC retention. Significance rests on the 6 biological replicates. Outputs by phase, the UMAP, marker heat map, per cell type DE tables and the cell type calls. View previews, download grabs one or all. The natural next move for single cell. The H5 export has an Open in Cell X gene button. Browse the annotated UMAP and expression live, or switch to RStudio. Clicking View on the UMAP renders it inline. 9,814 cells colored by 8 SingleR cell types, with a legend. Methods: paste ready, empty drops QC, scRAN normalization, SNN Louvain clustering, SingleR annotation, and aggregate then DECQ2 with versions and parameters. Parameters: phase, step, value, empty drops, FDR, HVG count, SNNK, the SingleR reference, the pseudo-bulk design. A ready supplementary table. Software pins exact versions, droplet utils, scRAN, scatter, SingleR, DESEQ2, the reproducibility baseline alongside parameters. Citations: Amiskita, 2020 for OSCA, Lun, 2019 for empty drops, Aaron, 2019 for SingleR, Love, 2014 for DSEQ2. QC audit logs every gate, cells retained, cluster count, and the replicate unit gate that enforces pseudo-bulk over per-cell DE. That's the single-cell thread. Route to OSCA, cluster, annotate in parallel, pseudo-bulk DE, a 7-tab report. One object, statistically honest DE.